This is a tutorial for the CRISPR GuideXpress web tool. We'll be going over how to search for orthologs between Drosophila melanogaster and Anopheles gambiae, as well as between other mosquito species, and then also how to search for mosquito CRISPR SGRNA designs. To see a list of the supported species, you can click here, and we have all the versions as well as a few example IDs. So to start out, I'm going to show how to map between a Drosophila melanogaster gene and Anopheles gambiae. You can pick the ortholog prediction tools. For this example, I'm just going to use them all, and the gene I'll be using is wingless. So we go ahead and search here, and this takes us to a table where you can see the gene you searched as well as the predicted orthologs. Clicking on either of these will link you to flybase or vector-base, and we give a score for the mapping. The higher score is a better mapping, and we show whether or not the mapping for this particular gene was the best score from fly to mosquito as well as from mosquito to fly, and then the ortholog prediction tools that were used and found this particular mapping. So I'm going to copy this gene that we found, and next I'll show how to map between mosquito species. So the first mapping I showed was based on these prediction tools. When mapping between species, we're using vector-bases orthology mapping. So just for an example, I'll map to Anopheles colutzii, and you can see the orthologous gene in this particular species as well. And then next, I'll just go over how to use the SGRNA search functions. So we give a few different options for how to do this. You can search by gene, location, as well as batch search with multiple genes or multiple SGRNA sequences. So for this example, I'm just going to do a basic gene search with the ortholog that we found in Anopheles gambiae. So this results page that you're taking to first shows a embedded JBrowse genome browser. Right now we're zoomed out, and it's showing us the gene as well as the location of exons, and down here you're seeing a bar graph with the number of guides in this particular region. And if we zoom in, so the easiest way to zoom in from here is just to double click. It's also possible to hold shift and then use the up and down arrow keys. But we can zoom in and see the SGRNA designs. If you click on one of them, it shows you some detailed information. We've got the genome coordinates, some efficiency scores, an off-target score, and a few other things there. If you want to get a better view of this genome browser, there's a full screen view button here. Clicking that will take you to this page, and you can see a little bit easier, and then there's also some on-screen buttons for zooming in and out if that makes it a little bit easier. Going back to the results page, if you scroll down, we also have a table with detailed results, and on here you can see an efficiency score, machine learning score, we have a seed score, off-target score. These are described here for more information, but we also have some other useful things like isoform coverage. So for a particular gene, in this case there was only one transcript, but it would tell you whether this guide would actually be cutting all transcripts for a particular gene. We also have the distance to the start codon, the percent coverage, whether or not the start codon was actually annotated, and then a few of these columns are specific to this species, but we're showing whether or not this guide will also work in Anopheles colutzii. And then if you click this link, it will take you to the scores specific to Anopheles colutzii. You can see it may have a different off-target score due to the other genome, as well as some other similar information. And then now I'll just show sort of a typical use case for how you might want to sort these results to look for optimal designs. So it's possible to sort this table by multiple columns by holding down Shift and then clicking on the column of interest. So for this example, I'll try and find guides that work in Anopheles colutzii. They're effective in wild populations, and then they don't have SNPs in the SWA5B cell line. And then I guess lastly, let's add off-target scores. So we'll get the lowest potential off-target score first. You can use these results just as they are, or if it's easier, you can export as either a CSV or an Excel file. And that's pretty much it. If you have any questions, you can contact us, and our information is here. We also have a bug report form. Thanks for watching.